Hello, everyone, and welcome to today's live broadcast. How can CellenONE technology help you automate cell line development? It is presented by Barbara McIntosh, the Vice President of Business Development at SCIENION, Alba Simon, Marketing Manager at SCIENION, and Guilhem Tourniaire, the Scientific and General Director at Cellenion. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen. Or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Barbara McIntosh, Alba Simon, and Guilhem Tourniaire. I will now turn the presentation over to them.
Thank you very much, Julie, for this nice introduction. So today I'm going to present you how CellenONE technology can be used to automate your cell line development. One crucial point in cell line development is the ability to isolate single cells. The main challenge in single cell isolation is to be able to dispense only single cells and to maintain those cells alive. And this has a number of applications. This application can be split in two groups. The group of single cell analytics, so where we want to analyze a single cell, and this is very useful when analyzing, for example, tumor tissue, where we know that there is a very large heterogeneity of the sample, and a single cell analysis will help you to obtain a more detailed and comprehensive picture of the cells that are contained in that tumor, as compared to a traditional method that would just give you an average value of the cell contained in that bulk. On the other hand, isolation of single cell is very useful whenever you're undertaking cloning steps, and this can be used for monoclonal antibody development, stem cell line development, and in application of gene editing, for example, using CRISPR-Cas9. One aspect that is very important when you're doing single cell isolation is to understand the aspect of Poisson distribution. Poisson distribution is actually a probability, and this probability gives you an indication of the likelihood for you to find a single cell in a given volume or output. When working, when doing limiting dilution, for example, which is the most common technique to clone your cells, people will typically work at a fairly diluted concentration in order to ensure that they limit the number of wells which will contain more than one single cell. Because, as you know, if you have more than one single cell, then you will need to reclone, so do a second step, which is a great loss of time. Over the years, people have developed a number of technologies to isolate single cells, and I'm now going to describe the different technologies that have been developed. So there is a number of technologies that are fast, such as the FACS, but they typically suffer from some drawbacks. So, for example, FACS is very quick. However, due to high shear forces, most of the cells that are coming out of a FACS are typically not alive or in poor shape. Then you have the technologies using the Poisson distribution, such as the automated liquid handler or microfluidic encapsulation. As described before, these technologies are typically very quick. However, only a small proportion of the well or microdroplet form will contain a single cell. On the other hand, you have some technologies that are more precise, but these are typically hard to automate and fairly slow. So this can be like laser capture, microdissection, or acoustic and optical tweezers. Another way of seeing those type of technology is on the plot. So we've plotted the precision versus the throughput. On the bottom right, you can see the technologies that follow Poisson distribution, microdrop, lead generation, traditional liquid handler. And on the top left, technologies that are precise but slow, so such as laser capture and acoustic and optical tweezers. The technology that we have developed is standing in between those, so it offers a very good precision while maintaining a fairly high throughput, and it is based on piezoacoustic dispensing and visual feedback. And now Barbara McIntosh is going to present to you the technology on which all this CellenONE was based.
Thank you, Guilhem. So to talk about the technology, piezo dispensing technology, I want to go back in time just a little bit. So Cellenion is an actual daughter company of our parent SCIENION. And Cellenion is focusing on the single-cell applications, whereas SCIENION is based in Berlin and is focused on ultra-low drop precision dispensing for a variety of applications in both the diagnostic and the life science marketplace. With this technology, we're able to dispense non-contact of a variety of different content, if you will, anything from DNA to proteins to particles, and as you can see, living cells. So within SCIENION, we used to dispense bulk cells at once to do specific applications, but as we got more specific in single-cell work, that's when we spun out our daughter company, Cellenion. With the technology as well, you can dispense to any type of support. So if you're doing nitrocellulose membranes or a slide or a microtiter plate or even a wafer, because the dispensing is non-contact, it's very much what we call an open platform. The benefits of dispensing non-contact, the obvious one is you don't physically come in contact with the surface. So you don't damage it like something if it's a coated slide or nitrocellulose. But also, and very critical when you're scaling, is the reproducibility. The reproducibility of non-contact dispensing is far superior than any other type. But with our technology, we have the droplets free-falling, and as they come in contact with the substrate and or other reagents, they have a very high efficiency of mixing. So with the technology, the piezo dispensing, piezo technology works by having a voltage applied to a crystal. And what happens is the crystal is squeezed and small droplets are ejected very quickly. Our technology allows us to dispense down in the picoliter range as small as 50 picoliters per drop, but it goes up to a range of 800 picoliters. And although that might seem very small, we dispense very fast, 1,000 hertz or up to 1,000 hertz, which means you can dispense 1,000 drops a second. So very quickly, we can get up into the nanoliter range, which is very attractive for single-cell dispensing. The next slide, you see actually an image. So this is an image of the piezo nozzle, and although it's sideways, you see a droplet being ejected from it. This is also a screen capture of the software where it calculates the drop volume. You can see in the corner it's 285 picoliters, but also very good quality control parameters like the CV of that dispense. And you can very quickly save these files as part of your quality control. And so these are one of the cameras that we use on our standard platforms, but we've also incorporated it into the CellenONE technology. This next image you can see, it's a video, and it's actually showing the droplet being ejected live time, but it is available on the SCIENION website. The second camera on the system is what we call target recognition. It's a CCD camera, and we use it a lot in diagnostic applications because it's registering a fiducial and it's moving relative to it to enable very precise dispensing to a location. And again, very, very beneficial for the CellenONE technology because once we see the cell traveling through the nozzle, we then move the dispense to the target position. This next video is showing a variety of different dispense applications, from anything to a small drop to patterning an area with an array. You see the task run going and capturing the drop in front of the stroboscopic camera, calculating the volume, and then moving and placing that drop to a different substrate. It's calculating standard deviation. You're able to pattern spots, change the volume and coat an area, or do a variety of different patterns. So as you can see from the video, this technology enables a variety of different applications. You can dispense into a microtiter plate and take a traditional ELISA and make it a multiplexed ELISA, allowing you more data with less volume of reagents. We can dispense with a microfluidic chip with blister packing, a transdermal patch, a wafer to do different types of diagnostics, because all of it is using the piezo dispensing. Sorry, my slide isn't advancing. So with the piezo dispensing, that's what we incorporated into the CellenONE, and now you'll be able to see some of the hardware and the software features that we use in our core technology and how we're enabling it for just single cell dispensing. So Guilhem, I'll give this back to you for the next segment.
Thank you, Barbara. So I'm just going to present you the different steps that have been taken to develop this new technology. So we start from a piezoacoustic dispenser, as described by Barbara, and we have this automated optical monitoring system. And basically, this work has been done around 2 years ago. It was initiated with a collaboration with the University of British Columbia. And there we started by studying the behavior of cells inside different geometry of dispenser. And by optimizing the geometry of dispenser, we could reduce the number of phenomena that could happen to only two phenomena, which we translate as travel or ejection. As you will see on the next slide, the size of the ejection zone, the green zone on this dispenser, is actually the area where whenever we have a single cell in that area, we know that this single cell will be contained in the next droplet that will be generated. Another thing we have to take into consideration is the fact that cells or any particles suspended in a medium with a lower density will tend to sediment. We implemented a sedimentation zone. This is to account for the fact that the cells will sediment in between the time we detect them in front of the camera to the time we go to the target. And basically, this gives rise to 4 different scenarios. The scenario we are looking for is to obtain a single cell in the ejection zone with no cell in the sedimentation zone. Whenever this is happening, we move the dispenser to the target and we dispense. The other 3 scenarios are: we have an empty ejection zone, so the droplet is discarded. Second scenario would be we have multiple cells in the ejection zone, then obviously we don't want multiple cells, so we will also discard this droplet. And finally, if you have a single cell in the ejection zone, but you also have a single cell in the sedimentation zone, by safety we will also discard this droplet. When I say discarding, one thing that is very important is that we discard those into a recovery tube, so the cells could be further aspirated and reprocessed if desired. And now you're going to get an animation that's going to describe to you the way the system, the CellenONE X1 system is working. So you've just seen how the CellenONE X1 works. So we aspirate a sample from a multiple well plate, for example, then go in front of the camera, undertake an automated mapping steps. From this mapping step, we determine the size of the ejection, sedimentation, and travel zone, and we start processing your sample. On the next slide, what you will see is actually what is happening on the software and inside the machine. So not anymore an animation, but real life. And this is an example that has been processed using prostate cancer cells. And on there, you will see on the right part is what is recorded by the software live, and you have a synchronized image on the left of what is happening in the dispenser. So whenever the image will freeze on the right, it means that the dispenser has detected a single cell, the head is moving to the target and dispensing a cell. So I'm just going to start it now. So what you just saw on this video was actually single cell being isolated and dispensed into microwells onto a microscope slide. And out of 400 position fields, 98% of the positions, 394, contained a single cell. And this was filled in about 18 minutes. One very important thing with this technology that people really enjoy is the fact that they can see live their sample being processed. Unlike a cytometer where you only get a signal, on the CellenONE you can actually analyze your sample, see if there are a number of aggregates, debris, and so on. And all these images are recorded and can be used for further quality control. And now my colleague Alba Simon is going to present to you the kind of results that have been obtained with this technology.
Thank you, Guilhem. So we have seen the background of the technology and how the gentle dispensing is able to maintain and isolate single cells. And now I would like to go into details of some of the results that we have compiled of the months from the machine, from the CellenONE. So just to recall what we mentioned at the beginning, by limiting dilution, which is following the Poisson distribution, we were fairly limited to 25% of single cell per drop, whereas with our technology, we are reaching up to 98%, up to 100% most of the time single cells. And that has been shown in these two nice arrays, where single cells from dissociated lung cancer spheroid were isolated into microscopic light, and that's the left side. And on the right side, we'll see the isolation of fluorescence microbead of 21 microns of diameter. And we see in both sides that we reach 100% of single cell in each position. We show us, first of all, is a highly precise system allowing the isolation of the single cell, and is also precise in the sense that we can reach very narrow positions, such as isolating cells in 50 microns microwells. In this picture showing on the top fluorescence microbeads and on the below part ELA cells. Now, we do several experiments with different type cells because we want to have a look if there is a different type that would be also as successful independently of the cell type. And we found out actually that we could reach a very high accuracy with fibroblasts, CHOs, and etc. A fairly high amount of different cells, and always finding single cell in each position. So we're jumping into one of the applications that we're focusing today, which is cloning. And we've already mentioned the main challenges of this application, which is, first of all, we want to find only single cells. And we, of course, want these cells to stay alive. So we have defined clonal recovery as the percentage of wells which will be containing single colony after an X amount of time. Sorry. Yeah. So just to shortly describe how does it look like when you are at the lab doing the cell line development, you will have to undergo, first of all, the genetic modification of the cell, then to do culture of 2 weeks, and then start with the cloning. On the left side, we see how's the process with the limiting dilution. You will have to do several steps and repeat these steps in order to obtain a high amount of best clones. And at the end, we have estimated that you will spend around 10 weeks. With our system, in a comparative experiment, we basically reach monoclonality after only 2 rounds of culture, after 2 times 2 weeks. So after a month, we will be able to have very good results in monoclonality. So we basically save time. We basically save as well media and consumables. So still having a very good outcome with this system. Then what we followed was an experiment where we, after the isolation of the cells, we culture for 7 days. And we found out that on average, 84% of the wells contain single cell colonies. In comparison with what we would have obtained by limiting dilution, so manual, would be limited to 25%. Ending up on having to do several steps because we will have multiple colonies after each step in order to reach the monoclonality at the end. Now, we mentioned that feature, and is actually something that it's very useful when doing cloning, recording all the isolation step itself, meaning that the user can follow up in live the trajectory of the cell from when the cell is in the capillary till it reaches its target. Meaning that you see on the left side the picture A1, a very clear cell, which will end up on an A1 well on your plate. So you have live data thanks to the camera, which is recorded prior dispensing, and that you can find after in the files, and then go back and check how was your experiment, just to document results. And as you can see on the below picture, it's very clear that in that case, a double cell, a doublet, will be dispensed, so therefore discarded in our recovery tube. Now, we did a last round of experiments with one of our partners externally, and we wanted to compare how our system, CellenONE, will behave compared to a fax. So we started the experiment. The aim was to clone and to look at only the negative cells, the GFP negative cells from a CHO cell media. Starting point was only 70% of this population was the population of interest, because only 70% were negative. So looking at what the fax can do, we know that the fax will sort, and we looked up only at only at the negative cell, meaning 100% of the cells will be negative GFP, whereas our system will look at the full amount of the cells, meaning that a maximum which is expected to be CFP negative are 70%. Well, then we did the culture media after 5 days, and we found out that actually with the CellenONE system, we reached this 76% of outgrowth, so viability cells which were alive, whereas with the fax only 38% were alive. And this is showing us that during the isolation process, the fax area 3 basically underwent high stress for the cells, which make them not to survive. In contrast with our system, CellenONE was ensuring that all isolated single cells were maintained alive, so reaching a fairly high percentage of viability. Now, a last comparison we looked at is, we know that our system is able to control the temperature of the target. So we basically did several experiments at 4 degrees, at 20 degrees, and 37 degrees, and we found out that cooling the target at 4 degrees was actually proving higher clonal recovery till 87%. So if you have to do a cloning experiment, we will recommend you to go to 4 degrees. Now, just to finish up with some of the results we recently got, was looking at isolation of mouse neural stem cells using our CellenONE. And as you can see in the picture, the results are already very promising. 97% of cells were single cell. Only 2% were empty, and only 1 was double. So just to summarize the main benefits of our technology, we will look at a system that is very versatile, which is accurate. It will isolate up to 100% of single cells. It will keep the cells alive, so keeping a very high amount of viability. It is an open system. You can use any of the consumables of your choice, and we are reaching a high throughput, a fast isolation process. As an example, 90 single cells were isolated in less than 4 minutes. So now in this presentation, we just focus on cloning, but as you know, our system has several other applications. And you can have a look on our website www.cellenion.com, where you will see several more information, more videos, and different applications of our system. Being said that, we thank you very much for your attention, and you have here our further information details, email and phone. And just to finish up with a little touch, the logo you see below is a live logo, meaning printed living cells. Thank you very much for your attention, and now we are open for discussion.
Thank you, Barbara, Alba, and Guilhem, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. The first question is, what are the overall benefits of your technology?
I'll take this one if you want. Guilhem speaking here. So we have a number of unique benefits from our technology. I'll try and summarize them quickly. But as you've seen, the technology is very accurate, which ensures that you obtain only single cell. Being very gentle, using this acoustic technology, we ensure very high viability. It is fully automated. It provides you with data that you can record, which are important for monoclonality, proof of monoclonality, so you can record images. We have temperature control, which allow an even more enhanced viabilities. And very importantly, it's an open platform, so you choose the type of consumable that you want to use.
Who is using your technology? Reference clients?
So, sorry, this is Barbara. So the technology itself has been in the marketplace for a variety of different applications, from, as I mentioned earlier, life science to diagnostics. And you can see some of our clients on that slide from both academia to full-on commercialization, because the technology is scalable. Can you click? Now, specifically to the CellenONE technology, we've collaborated with a number of clients to enable both software development and proof of concept applications using a variety of different cells and development.
Which cells or particles were successfully isolated with CellenONE technology?
So here's Alba speaking. So basically, we have reached very good results on isolation any particles or cells from 1 to about 40 microns diameter. These cells are, the major group are mammalian cells, going from B cells, CHO, EGK, hybridomas, neural stem cells, lymphocytes, tumor cells, CTCs, hepatoblasts, and primary fibroblasts, for example. But we are also undertaking proof of concept for cells, including bacteria cells and yeast, for example. And we have, of course, dispenses barcoded normal beads used in sequencing, like the McIntosh beads, for example.
I would like to once again thank Barbara McIntosh, Alba Simon, and Guilhem Tourniaire for their presentations. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through July 12, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.